peculiarity, or say one of the very important feature of the histidine operon. The another very important feature, as you know, is the presence of 10 structural genes. There are 10 structural genes, and the peculiarities as far the structural genes are concerned. These 10 structural genes encode only 9 enzymes, and these 9 enzymes catalyzes the 10 steps involved in the biosynthesis of histidine from its precursor. It means that the two structural genes same enzymes. So there are 10 structural genes encoding 9 enzymes which are required for catalyzing the biosynthesis of histidine from its precursor. So this is another very important feature of the histidine operon. Beside this, you know that there is different opinions regarding the location of the promoter gene. This is also very important feature. The location of promoter in the histidine operon is not very clear. It is not yet known whether it is located in between the regulator and operator genes, or whether it is incorporated or included within the regulator genes. So it is not still clear. So this is very important. This is also very important feature, that the location of the promoter is not well defined in case of histidine operon. In addition to these three peculiarities, you know that the histidine in this case, like the tryptophan operon, acts as the co-repressor, and attenuation can also take place in this histidine operon like the tryptophan operon, because it is also a repressible operon. And you know that like the tryptophan operon, this histidine operon is also negatively controlled operon. Positive control is absent in this case. So these are some of the very important distinguishing features of the histidine operon. Now today we are going to discuss the functioning of this histidine operon. Okay? How this histidine operon functions. So like other operant, you know that there are two cases: switching on of the any operand and switching off of any operand. So this histidine operand gets switched on when there is low level of histidine in the bacterial cell. What happens, as you know, that the regulator in case of the histidine operon encode inactive repressor, and this inactive repressor, as you know, is known as the aporepressor. So this inactive repressor encoded by the regulator gene in case of the histidine operon is inactive, and it is not capable of binding at the operator site. So if the operator remains free, what happens? If the operator remains free, RNA polymerase will bind at the promoter region, and it will initiate the transcription of the structural genes, 10 structural genes, and these 10 structural genes are transcribed on a polycistronic mRNA. You know that the meaning of the polycistronic mRNA. You know that if more than one structural gene is transcribed on any mRNA, so this is called polycistronic mRNA. And in case of prokaryotic organism, the more than one structural genes are transcribed on the same mRNA. So in case of prokaryotic organism, mRNA is polycistronic. But in case of eukaryote, like human being or any other eukaryotic cell, the mRNA is monocistronic, because only one structural gene is transcribed on a particular mRNA. Not more than one structural gene can be transcribed on the same mRNA in case of the eukaryotic cell. So this is a very important difference between the transcription of the in prokaryotes and eukaryotes. Okay? And you can write that if you are asked to distinguish between the prokaryotic and eukaryotic mRNA, so you may mention different types of differences among them, between them, but this is also one of the very important difference between the prokaryotic and eukaryotic mRNA. Prokaryotic mRNA is polycystonic and eukaryotic mRNA is monocystonic. So just you have seen here that the 10 structural genes in case of histidine operans are transcribed on the same mRNA, constituting a polycystonic mRNA, and this polycystonic mRNA undergoes translation to produce the enzymes which are required for catalyzing the process of biosynthesis of histidine from its precursor. So in this condition, when the inactive repressor does not bind at the operator site and promoter becomes free and transcription starts. So in this situation, the histidine operand is said to be the switched on histidine operand. Now let us discuss the second case. When the when when the level of histidine is higher in the when the level of histidine is higher in the bacterial cell, what happens? The histidine acts as co-repressor. You know that in case of repressible operon, like tryptophan operon, the tryptophan also acts as the co-repressor. Similarly, in this case also, the histidine itself acts as the co-repressor. So when the level of histidine is higher in the bacterial cell, the histidine acts as the co-repressor, and it inactivates. And this histidine, when the level of histidine is higher, what happens? This histidine brings about the activation of the inactive repressor. You know that the repressor in this case is the inactive, and it is not capable of binding at the operator site. What happens? This inactive repressor is activated. This inactive repressor is activated by the co-repressor, which is histidine in this case. So histidine acting as a co-repressor brings about the activation of the inactive repressor. And when the repressor becomes active, when the inactive repressor is activated by the co-repressor, which is histidine, the active repressor now becomes capable of binding at the operator site. And when this uh, active repressor binds at the operator site, that is when the operator, as a result of which the operator gets blocked. And when the operator gets blocked, RNA polymerase cannot bind at the promoter site. And if there is no if the binding of polymerase at the promoter site does not take place, there is no question of any transcription. The transcription stops, and there is no further synthesis of the enzymes meant for catalyzing the biosynthesis of tryptophan from its biosynthesis of histidine from its precursor. So when the level of histidine is higher in the bacterial cell, this histidine acts as the co-repressor, and this co-repressor brings about the activation of the inactive repressor. And when the inactive repressor becomes active, it binds at the operator site, and it prevents the binding of promoter at the it prevents the binding of polymerase at the promoter region. And in this situation, no transcription can take place. And if this is the situation, the histidine operand is said to be switched off. The diagrams representing these two cases have already been drawn on the whiteboard, which I would like to share with you so that you can easily understand it. So now look at the whiteboard and try to understand. Is the whiteboard visible, Tustika?
Yes, sir.
This is the case. This is the case one. Case one, switching on off histidine. I have shown here. Look at the whiteboard. Who is making noise? Let me know whether it is visible or not. Look here. This is the histidine operand. I have shown here the regulator genes RUST, and this is the W. So these five regulator genes represented by alphabet RUST and W has been shown here. And there are two cases. Either the promoter is located in between the regulator and operator, or it may also be located in intercalated among the regulator genes. So it doesn't matter whether it is located in between the regulator and operator, or it is present within the regulator genes. And this is the operator genes, and these are the 10 structural genes. What happens? This regulator gene encode the inactive repressor, which is known as aporepressor. The aporepressor has been shown in the red ink here. This represents here the aporepressor, and this aporepressor cannot bind at the operator region. Operator remains free here. Therefore, the operator remains free, and when the operator remains free, what will happen? RNA polymerase will bind at the promoter region, and the transcription will start. The transcription starts, transcription takes place, mRNA is formed. This mRNA undergoes translation to produce the proteins, and these proteins act as the enzymes, and these enzymes catalyze the biosynthesis of histidine. So in this situation, the histidine operon is said to be the switched on. Now look at the case 2. This is the case 2. Switching off of histidine operon. Look here. This regulator gene encodes the aporepressor, which is inactive, which has been shown in the red ink. Histidine acts as the co-repressor, which is also shown in the red ink here. So this histidine brings about the activation of this aporepressor. I have shown here the active repressor and represented the active repressor in the green ink. This active repressor is capable of binding at the operator site. Now look here, at the operator site, the active repressor has bound. Okay, and now this operator has been blocked. Okay, due to the binding of the active repressor at the operator, RNA polymerase cannot bind at the promoter region. RNA polymerase now cannot bind at the promoter region because the operator is not free. What happens? No transcription will take place. There will be no transcription because operator is blocked, RNA polymerase is not binding at the promoter site, and in this situation, there will be no further transcription of structural genes. There will be no biosynthesis of the enzymes, and in this situation, the histidine operon is said to be the switched off histidine operon. So there are two different conditions of the, and I think that